Australia is a huge country covered in an expanse of forest, coastlines, and grasslands. The produce we eat are grown under a variety of systems and are minimally processed after post-harvest. Once packed, they can be transported over different distribution pathways where they can then become exposed to different conditions that may influence product quality before reaching consumers at the shops. But what about food safety? Fresh produce can become contaminated with harmful bacteria during production, at harvest, or during post-harvest processing, such as workers handling produce or due to contaminated equipment. If produce are then exposed to varying conditions during storage and transport, this may provide the opportunity for these harmful pathogens to grow and/or survive. And if they grow to such high populations, this can result in foodborne illnesses or worse, as outbreaks as we have seen over the last decade. So, we wanted to know which conditions may influence this growth during storage and transport. We used Listeria monocytogenes contaminated on whole fresh cucumbers, which were simulated over 2 real-life supply chains in a laboratory setting based on real monitoring data. We found that a high dynamic range, a 14-degree difference between 9 to 23 degrees, resulted in pathogen growth, whilst a lower dynamic range, a 5-degree difference between 9 to 14 degrees, did not support pathogen growth over 2 days of storage. We also saw that lowering the humidity by around 20%, from 97 to 78%, also resulted in higher pathogen die-off on cucumbers. When we pushed conditions to really abusive temperature levels, at 40 degrees Celsius, our results showed that Listeria monocytogenes growth was also not supported on cucumbers. This was an interesting finding, as previous studies have observed that Listeria survives or can decline at 5 degrees, but storage temperatures greater than 10 degrees resulted in growth. It is also important to note these studies sampled on sliced cucumbers and not on whole. Listeria is known to survive and grow slowly at temperatures from 5 degrees and grow optimally between 30 to 37 degrees with a maximum limit of 45 degrees Celsius. Other conditions for optimal growth include a pH environment between 6 to 8 and water activity of 0.97. Cucumbers have an optimal storage temperature of around 10 to 13 degrees with a pH level of around 5.5 and water activity of 0.99. So these conditions are within range for which Listeria monocytogenes should be able to grow. So we thought to ourselves, why was that not the case? We think that specific characteristics of the cucumber surface plays a role. One possibility is that the natural microbiota on the cucumber surface may outcompete pathogenic Listeria populations during storage. So what we did was delve into metagenomics. To put it simply, it is a genomic analysis of microbes by extracting and cloning DNA from a microbial community. In our case, the bacterial communities on the cucumber surface contaminated or not with pathogenic Listeria, which we then stored over 2 weeks at optimum temperatures. This huge amount of information allows us to capture the full spectrum of microbial diversity, which traditional culturing and plating methods cannot do. And what we found is that for the same commodity, there was no difference in bacterial communities whether cucumbers were contaminated or not with Listeria, nor any difference through the 14 days of storage. What differentiated the cucumbers was its place of origin. From 4 cucumber suppliers, 2 originated in New South Wales, 1 in Victoria, and another in South Australia. Some species were unique between each of these suppliers, but what was most interesting are the microorganisms similar between them. For example, Pseudomonas was found across all 4 suppliers, and studies show that they compete with other organisms and produce a wide range of antimicrobial compounds. And Paenibacillus was found in 3 suppliers and is known to have inhibitory functions against Listeria monocytogenes. Our studies have shown that supply chains can be highly variable within a commodity group and across regions. Other factors such as community composition, duration of the supply chain, temperature, and relative humidity also plays a role on pathogen behavior on produce. We know for sure that it is crucial to monitor the storage and transportation stage as to minimize the microbial risk involved in the distribution chain, especially dynamic temperatures which exceed optimal cucumber storage conditions. If we take into consideration these factors affecting pathogen survival, then we will be able to improve current risk assessments and ensure the safety of fresh produce to consumers. Thanks for listening.